Today we're going to talk about Kingdom Monera or Kingdom Prokaryota. The reason it's called Kingdom Prokaryota is that it is essentially totally filled with animals that are prokaryotes. As you remember, the prokaryotes are those cells that have no membrane-bound organelles. So these are very simple organisms that function on a very basic level. When we talk about the Kingdom Monera, there's a couple things that are sort of separate Kingdom Monera from the other kingdoms, Kingdom Animalia and Kingdom Planta. And one of those things is the fact that as organisms go, these organisms are not well understood by biologists. They have a lot of functions and a lot of structures that are still, it's not clear what their purpose really is. The other thing that's even more interesting, at least to me, is the fact that these organisms can survive in environments that for most organisms would be deadly. There are organisms that can survive at very high temperatures, temperatures in which no normal organism would survive. They can survive at very low temperatures. They can survive in environments of very high salt concentration. They can survive in environments with no oxygen. They can survive in environments that have a great deal of oxygen. So the Kingdom Monera, there are organisms in that kingdom that survive on what I would call the edge of environment. The areas where other organisms cannot survive. You'll find as we talk about other organisms that most of them exist in a very narrow band of conditions that have to be almost absolutely perfect for them to survive. The Kingdom Monera is occupied by a group of organisms that are not quite so picky in terms of their environments. Kingdom Monera has primarily bacteria, and bacteria are single-celled organisms that essentially make up all of the kingdom that we're talking about. Now, bacteria naturally are thought of in terms of their ability to cause disease, and these are all just examples of diseases that can be caused by bacteria. This is an area, an abscess area in this person's leg. These are two examples of cellulitis where there's an infection in the skin here. But actually, bacteria can be bad, that is, cause disease, and in which case they're called pathogens. But they can actually also be good. The intestinal tract of the average human has approximately 10 to the 10th bacteria in it. And so it's very common for bacteria to exist in relationship with other organisms and actually be necessary for that organism to function. Now, as we talk about these organisms, we're going to talk about some of their specific structures. One structure that many of these organisms have, now not all, but many, is called the capsule. Now the capsule is composed primarily of carbohydrates, and in this case these carbohydrates are termed, quote, sticky, end quotes, and the capsule is important for the bacteria to adhere to other substances. Now sometimes that adherence is related to surfaces, other times that adherence is related to sticking to cells within another organism's body. Sometimes the adherence is sticking to each other. But frequently this capsule is important for the organism to stick to something else. Now, actually, in some cases, the capsule can either be sticky or smooth. And the smooth capsule actually makes it more pathogenic because the inflammatory cells cannot stick to it and destroy it. Another structure shown here is called the cell wall. Now, the cell wall, as we know, is seen in plants but not animals. It's also seen in bacteria, and it forms several functions for the bacteria. First of all, the bacteria doesn't have any other way to do this, and so it holds the bacteria together. Secondly, it helps determine the bacterial shape. Round bacteria are called cocci. Rod-shaped bacteria are called bacilli, or bacillus. And then lastly, the cell wall functions to regulate water distribution inside and outside the cell. The third part of the outer aspect of the bacteria is also present in other organisms, and it's the plasma membrane, and we've already talked about this at length. One other thing I'd like to say about the cell wall is this is one of the areas where a lot of our antibiotics that we use attack. They take advantage of a weakness in the cell wall and cause problems with the bacteria's ability to control its external environment. As I was saying, with the plasma membrane, it in many ways is similar to what we've said about plasma membranes in the last lecture. It's responsible for transport and controlling molecular substances inside and outside the cell. Now this points to, it's not supposed to be pointing to that dot, it's actually supposed to be pointing at the white substance in between the dots, which is called the cytoplasm. The cytoplasm is, we have talked about before, and it's the fluid substance that's throughout the interior of the cell, in which all the other stuff that's inside the cell float around in. This winding, sort of snake-like looking structure in the middle of the cell is the bacterial DNA. Bacterial DNA is not bound by a membrane, and therefore is not in a nucleus. It holds the genetic material for the cell. Now, bacteria actually have an extra part of DNA that's not part of this central portion. It's not shown in this diagram, and that extra DNA is called a plasmid. But this DNA is important because it codes for all the information in the cell, and when the cell replicates, this DNA has to be replicated as well. And then these little dots that are sort of scattered throughout the bacteria, they are ribosomes. And as we know from our previous lecture, ribosomes are important for making proteins. They are the factories in the cell that make the proteins that the bacteria uses to perform certain functions. Now, when we look at these projectile structures outside the cell, first of all, we have these long projectile structures. Now, many organisms in this kingdom have only one of these. This one actually shows four, which I think is actually somewhat unusual to have several of them like this. But this is called a flagella. That's the plural. Flagellum is the singular for that word. And it's large, long, whip-like structure whose primary function is to help the bacteria move throughout its environment. The second structure, which I did not point to because I didn't want it to be confused with the capsule, are called cilia. Now, not all bacteria have cilia, but those that do, they are hair-like projections that come off the cell wall, and so they would look like pretty much in the area of what's shown as the capsule here, and these are also important for motion. Now, whereas the flagellum has very large sweeping motions that can easily be seen, the cilia beat in a sort of rhythmic fashion together like the oars on a galley boat in a Roman movie. They all beat together to help the cell move throughout its environment. The third structure, which I have circled here, is called a fimbria. Now, fimbria, the way it's spelled here, again, that's plural, and these are more short, stubby projections, and essentially they're helpful for the bacteria to hold on to things. And so basically we see that these structures are used in order to hold on to another bacteria, in order to hold on to the surface of its environment, in order to anchor the bacteria when it doesn't want to move. Now, there's a subtype of this fimbria that is responsible for holding bacteria together during reproduction, and these are called sex pili. And these are basically necessary to help the two bacteria, and we'll talk about how this occurs, but the two bacteria come together, they are held together by these fimbria, and then there's an exchange of DNA between the two bacteria. Now this diagram is an enlargement of a flagellum, and the flagellum can be separated into three parts. The first part is circled here. This part is called the filament. The filament is the long portion of the flagellum that does most of the flapping that you see when you look at this under the microscope. And so when there's a whipping back and forth, what's whipping back and forth is the filament of the flagellum. The second part, shown here, is called the hook, and the hook essentially attaches the filament to the basal body. It's a short L-shaped structure, and you should think of this as turning in a circular manner, and then the filament then whips in a sort of circular manner like a propeller. The third portion, circled here, is the basal body, and the basal body is where all the work is done. The energy is expended here, and it generates the actual motion that's transmitted to the hook, that's transmitted to the filament. The best way to think about this is to think about it like a motorboat motor, in which the basal body acts as the motor, the hook acts like the center part of the propeller that's attached to the motor, and then the filament is the wings or the blades on the propeller. The motor turns in a circular manner, causing the central shaft of the propeller to turn, and then it results in the turning of the blades, and then the boat moves through the water. This is exactly how a flagellum works. There's about 30 proteins that interact in the basal body with exchanges of energy that lead to the rotational motion. This is transferred to the hook, which rotates, and when it rotates, it causes the filament to rotate and propels the cell through the body.